Now the question is, how do these signals actually control and engage the proteins? We recognize there are two parts. Yes, a signal and a protein. The protein are the components inside the cells that carry out the functions of the cell. The signals, by their nature, come from the environment. Well, the interesting thing about this is that the proteins are on the inside of the cell, and the signals that drive these proteins are actually derived from the external environment. So the issue is, how are the signals picked up by the proteins? Because inside a cell, they're encased within a cell membrane and buried within the cytoplasm. The question is, how can an environmental signal get from the outside and get to the inside to control the functions of the proteins? Well, this is where my work came to a very exciting point way back in the early 80s. I started to recognize, my goodness, the membrane, the skin of the cell, is an interface between the external world where the environmental signals are derived and the internal world where the proteins lie. And that for the proteins to be activated, the signal has to cross through this membrane. So I started to take my attention and apply it to the understanding of the structure and function of this membrane. To just give you a little insight into the character of the membrane, the skin of the cell, let me give you this point. The skin is so thin that we can't see it in a regular microscope. And in fact, until the 1940s, biologists were not even sure that cells were even surrounded by a skin. Many scientists actually thought cells were like little jello moldings with fruit compote in the inside of it, where the fruit were the organelles, and the organelles were suspended in a gelatinous matrix. Yet others believed there was indeed some kind of membrane surrounding the outside that would contain the cytoplasmic components. It was not to the development of electron microscopy that we were able to actually resolve the structure of the membrane, because it's so thin it's below the limits of resolution of the light microscope. You just can't see it. But once electron microscopy started to observe membranes, they found them everywhere. That every cell from every organism, from the most primitive organism on the planet, such as bacteria, to humans, all cells have cell membranes. And another interesting fact, all cell membranes appear the same in regard to their structure. And that was a real interesting recognition because, while many organisms have different kinds of components in their cytoplasm, some expressing, let's say, chloroplasts, such as plant cells, and animal cells not expressing those. Animal cells having hemoglobin, and that's not present in plant cells. That all different species had different kinds of components. However, there was one uniform character. Every living system had a surrounding, controlling membrane, a membrane that enclosed the cytoplasm. This membrane acted as an interface between the external world and the internal world of the cell where the proteins reside. The other interesting thing about the membrane that it was found it can be comprised of two basic components. One component are little molecules called phospholipids. As the name says, lipids mean that they have an oil characteristic to them. The actual molecule actually has a water-loving and oil-loving component in the same molecule, in the sense that molecules normally that dissolve in water are water-loving molecules, and molecules that dissolve in oil are oil-loving molecules, and oil and water don't mix. Now I'm talking about a molecule that is comprised of both oil and water. And so in a sense, it doesn't know whether to dissolve in water or to dissolve in oil. However, when thrown into water, these phospholipid molecules will spontaneously self-assemble into a layered structure. They actually will automatically form a membrane. As the lipids try to respond to the water, they create a layered structure in which the membrane lipids, the lipids component of the molecules, are found in the center of the layer, and the phosphate heads of the molecules, which love water, are found on either side of the layer. So the membrane has a three-layered structure. In the electron microscope, when you see it stained, it's black and white. There's a black layer, a white layer, and a black layer. In a sense, it almost looks like an Oreo cookie in the electron microscope. And it turns out that the icing inside the cookie is the equivalent of the lipids. Since oil and water don't meet and match with each other, since they separate from each other, then this membrane creates an interesting barrier or skin, which has a complete lipid, hydrophobic, water-hating center that surrounds the cell. So the cell is actually encapsulated in a barrier that contains a hydrophobic zone. But this becomes very important because now this membrane acts as a skin. It keeps the components from the outside world from readily entering inside the cell, and it keeps the components from the inside world from readily escaping from the cell. And that is because the little hydrophobic layer in the center of this three-layered membrane is a barrier that prevents anything that's soluble in water from going from one side to the other side. So the membrane is an impermeable skin. However, a membrane just made out of phospholipids would not really support life. Because if a cell had a skin that wouldn't let anything in and wouldn't let anything out, the cell would not survive for the simple reason is how would it get nutrients to activate its biology? How would it get rid of waste products that were the result of metabolism? Since the membrane is an impermeable barrier, then these functions would not be able to occur. This is why the membrane has a second component. It has proteins. Proteins, again, those complex molecules, reside in this three-layered structure. The proteins are so large that most proteins actually extend from both sides of the cell. So when you look at a cell membrane, you actually see some parts of proteins extending to the outer side of the cell membrane, and the same protein actually, because of its size, extends to the inside. The proteins span the membrane, and as a result, the proteins provide for a conduit to allow things from the outside to use the proteins as a conduit to get to the inside. So the proteins act as a device to communicate between the inside and the outside of the cell. Now remember, we talked about proteins as these structures that can change shape. Well, as these proteins are built into the membrane, I could also tell you this. There are two primary classes of proteins built into this membrane. The two classes of proteins represent one form is called receptors. The other form of proteins are called effectors. Because these proteins are not resting on the membrane, they're actually directly incorporated into the laminar structure, the layered structure of the membrane. We refer to these proteins as integral membrane proteins, which is abbreviated as IMPs. There are two classes of these IMPs as mentioned, receptors and effectors. And here's how they work. The class of proteins called receptors are tuned to signals that are found in the environment. So a sense, like we mentioned before, a protein could be the shape of a baseball glove that would catch a signal that was the precise shape of a baseball. Well, consider that a protein in the cell membrane has the shape of a baseball glove. And if there's baseballs in the environment, the baseball will plug into the glove. But, as I also mentioned in that story of a signal binding to a protein, when the baseball plugs into the glove, it changes the charge of the protein and the protein changes shape. So, this baseball glove, which is extending from the surface of the membrane and catching a baseball, also extends through the membrane. And so when the baseball is received, the signal from the environment is identified and binds to the baseball glove, the protein changes shape. And inside the cell, you can see that protein changing shape because the protein of the baseball glove actually moves. And when the protein moves, it means that the glove has caught onto the signal. So if I'm inside the cell, I can tell you if a baseball is outside the cell by watching the receptor protein that has the shape of the baseball glove. Because when the signal, the baseball, shows up, the glove will change shape. Well, now we have an understanding of how a single receptor protein might work. Let me inform you of this. There are thousands and thousands of different kinds of protein receptors built into the membrane. The receptor's function is to read the signals of the environment. Different kinds of receptors read different kinds of signals. Some receptors read calcium. Some receptors identify glucose. Some receptors specifically match with estrogen. Other receptors specifically complement adrenaline. Some see histamine. Basically, whatever the cell can see in the environment, the reason it can see it's there is because there's a receptor that will complement with that signal. And when that signal shows up, that receptor will change its shape. And that will signal the fact that the actual signal is in the environment. So the changing of shape of a protein is a physical reflection of the presence of a signal. Well, this is very interesting, and it might seem a little bit obtuse. What is this membrane skin receptor business? Well, let me give you some interesting fact that we'll expand on in a little bit, and that is this. When I was teaching this cell biology and teaching about the proteins in medical school, in the opening day of teaching the course in cell biology, what I would inform the students of is a very important fact that most people weren't aware of. And that important fact is this. While you see yourself as a singular entity, the truth is you are not a single entity, that you are made out of living cells. It's the cells that are the living units that collectively make you. So you are actually a community of upwards up to 50 trillion cells. Secondarily, when you look at all the attributes that a human expresses, all the different kinds of physiological mechanisms and characters that humans express in their normal behavior in their normal lives, here's an interesting fact. There is not one new function present in the human that's not already present in every single cell. Wherever you have a function and an organ to carry out that function, this little tiny miniature cell has an organelle, meaning miniature organ that carries out the same function. So the idea is, if you really want to study the cells, you can look at a human and get parallel understanding because they're built in the same image. In fact, the human is made in the image of the cell. And this is why science can study cells and apply that information directly back to humans, because we share the same functions, we share the same structures, and that whatever we can do as a human can only be done because our cells are doing it in the first place. So with that parallel that I said we can look at a cell and look at a human and compare the two and make an understanding from it, let's go back to that cell. And I was talking about on the skin of the cell. The skin reads the environment because on the skin are receptors. And then I say, well, wait, you're made in the image of that cell. Guess what? You have a skin. It reads the environment. And you have receptors built into that skin. There's some very big receptors that are very obvious. Eyes, ears, nose, taste bud. These are obvious receptor structures that are built into our skin, and their function is to what? Read the environment and then send a signal internally so we can interpret and respond to the environment that we live in. The parallel is exactly the same. The surface of the cell is a skin. It has receptors built into it. The receptors read the environment, and the information of the environment is relayed to the inside of the cell so the cell can know what's going on. So now we've covered the first part of the membrane's function, reading environmental signals. Again, so does your skin. Not only do we have the receptors in the head, the very large ones like ears, eyes, nose that we talked about, but all over your entire body, the skin is filled with receptors to let you know what the temperature is, to let you know if something is touching you, to give you all information about the environment, let you know about painful things. All these are receptors, actions that are built into the membrane called the human skin. The same thing occurs, but at a smaller level in the cell. So now we have one set of proteins that we described. These proteins are the receptors. They read the environment and change their shape when the signal shows up. But that only tells the cell that a signal is there and identifies what signal it is, because the protein that responds will determine what signal was present in the environment. But now, how does the cell respond to the presence of the signal? How does it engage in a behavior? And the answer is the other set of proteins that are built into the membrane, the other IMPs called effectors. As the name implies, effector proteins create an effect. And so when these proteins are built into the membrane, they have, like all proteins, a resting conformation. But the effector proteins are coupled to respond to activated receptors. So in the surface of the cell, there are couplings of receptor and effector proteins. When the receptor is not activated, the receptor and the effector protein don't actually communicate with each other because the shapes or conformations of the proteins in their inactive state do not provide for coupling. But when the protein receptor reads a signal, remember, it changes its shape. So in a sense, the activated receptor has a different shape or conformation than the inactive receptor. The new activated shape it takes on turns out to be a physical complement that will plug into the effector protein, which was just resting there. So basically, let's review this. There are sets of proteins in the membrane that consist of receptors and effectors. The receptors read environmental signals. Before the signal shows up, the effector is in conformation A, the receptor is in conformation A, neither of them plug into each other. However, if the receptor sees its signal, the consequence of a signal binding to a protein changes the conformation. So receptor A changes its shape into conformation B. Yes, but B is a structure that is recognized by the effector, so the receptor now plugs into the effector. But once the receptor binds to the effector, it changes the shape of the effector because the receptor acts as a signal and changes the effector from conformation A to conformation B itself. But here's the important part. When the effector changes its shape, the response is it leads to an activation of a cellular function, such as respiration or digestion or movement of the cell. So basically, we stand back and we look at the surface of the cell, and it's filled with all kinds of proteins built into that membrane. And there we can see one pair of proteins, a receptor and an effector. And they're not connected because there's no signal present. And now we'll play through the system. A signal shows up in the environment that the receptor can recognize. The system leads to the receptor binding the signal. When the signal is bound, the receptor changes its shape. In the new conformation, the receptor then plugs into the effector. It couldn't do so before because conformation A wasn't recognized by the effector, but the activated form of the receptor is. So the activated receptor plugs into the effector, which in turn activates the effector to change shape, which in turn sends a signal into the cell to engage one of the functions. The function that engage is not a random function. The function that is engaged is directly related to whatever the signal is that showed up, so that the biology responds to the signal. To give you a personal example of what I'm talking about, consider this, that you walk out of your house and it's a very cold day. Your skin is your membrane. It's got receptors in it. You have receptors that read the temperature. When the receptor recognizes that it's cold out, it changes its conformation, sends a signal inside the cell, and that signal is mediated by an effector that responds to the sensory receptor. And that effector causes us to raise our metabolism and as a result generate more heat and keep our body at a constant 98.6. And basically, how did that work? A receptor at the skin picks up an environmental signal, engages an effector, which then adjusts the metabolism. So basically, the skin of the cell, the skin of the human are operating under the same mechanisms. Well, at one sense, somebody out there in the audience might be saying, now wait a minute. You said that humans and cells are sort of physical images of each other. In fact, that is true. Then I also said that the organs of the human are reflected in the organelles of the cell. And lastly, I said that the cell membrane, the organelle that surrounds the cell, is the equivalent of the brain. And then I said the cell membrane is the equivalent of your skin. That's where some of you might go, wait a minute. Are you saying my skin is my brain? Yes, in fact, I am. But this is obvious information if you understand embryology. And just to connect these to show the relationship between cells and human bodies, let me just briefly review the early stages of embryology. In the formation of an embryo, a sperm and egg come together, and the fertilized cell is called a zygote. The fertilized cell begins to divide and form a ball of cells. The first form of the embryo is this loose aggregation, a ball of cells called the blastula. But the next phase of development of the embryo involves a sorting out of the cells to form 3 concentric layers like a bullseye target. The layers in Latin are called derms. Derms means skin. That's why we talk about dermis and epidermis. So there are derms. There are 3 derms or layers of the embryo. The 3 layers from the outside in are called ectoderm, mesoderm, and endoderm. Ecto means outer, Derm means layer. Ectoderm, outer layer. Meso means middle. Mesoderm is middle layer. And endo means inner. And endoderm, therefore, means inner layer, the three concentric layers. What's unique about the layering of the embryo at this stage is that these specific layers give rise to very, very specific organs and tissues. And here comes the catch that's really fun. The outer layer of the embryo, called the ectoderm, only gives rise to two structures in the developed human. The ectoderm provides for the skin, and the ectoderm provides for the central nervous system. So the parallels of comparing the skin of the cell to the brain is the exact parallel of looking at the human embryo and its three-layered configuration, recognizing that the ectoderm provides for both the skin and the brain at the same time because they are connected to each other. So in this case, we go back to the cell and say, okay, how does the cell control its behavior? And the answer is there are IMPs built into the membrane of two classes, receptors and effectors. That receptor-effectors form combinations, where the receptor is responsible for identifying an environmental signal, and the effector is responsible for recognizing that the signal is present and then sending a signal into the cell to engage a protein mechanism that will make the appropriate behavior in response to that environmental signal. These proteins constitute switches. And how do they represent a switch? Well, look at it this way. The signal would directly drive the protein, but the protein is inside the cell, and the signal cannot get to the protein unless it crosses through the membrane. So the membrane acts as a barrier, but with these switches can allow specific signals to be recognized and the information sent through the membrane to engage the proteins on the inside. So when looking at the cell membrane, you're actually looking at the basic mechanism by which the brain operates, be it the brain of the cell or the brain of the human. It's exactly the same because of the relationship between cells and the structure of the human. Well, these are the switches then. And on the surface of the cell, there's not just a single switch. On the surface of the cell, there are 100,000 different switches. The switch is responding to everything that's in that cellular environment. So that the surface of the cell is a very complex structure. Not that any particular switch is complex. It's complex in regard to the numbers of switches that are simultaneously operating in a living cell. While I can easily sit here and describe the action of one signal affecting one switch, which in turn affects one behavior, human complexity comes in the regard of handling 100,000 switches and the consequences of where each switch is being on or off at any particular time. Yet, the fundamental nature of any switch and the mechanism it controls is very, very simple. So it's really interesting because in our effort to study human behavior and study the human brain, we're completely at a loss. 100 years of neuroscience and we really don't know how the brain works. The problem is the complexity of dealing with a structure that has 1 trillion cells. How can you really try to figure out the interactions among 1 trillion cells? This is why studying a single cell is very important, because by observing a single cell, I can see the mechanisms down at its lowest level of complexity. And when you start to see the basic nature of how a single cell works, you can take this work and apply it to the human. So we have at the surface of our cells switches. Now, when I first started talking about the switches to people that would attend lectures, I called them what they were. I called them IMP switches comprised of receptors and effectors. And I could see, obviously, that really tickled everybody's fancy because as I provided the name of that switch in scientific terms, everybody just looked at me with staring eyes going, what's he talking about? It's one of those complex scientific terminology things again. And I realized that really wasn't an adequate way to describe the switch. So I started to look at describing the switch by defining its function. And the definition of the switch is easily derived by defining the function of the receptor and defining the function of the effector. The receptor, the function is very obvious, as you're familiar with it yourself. It's awareness of the environment. That's what your receptors provide. Your eyes, your ears, your nose, these are providing information to you about what's going on in the environment. The protein receptors on the cell membrane provide to the cell information as to what's going on in the environment. So it provides awareness of the environment. The effector does something different, though. The effector sends a physical signal into the cell to engage any of the physiological mechanisms that would be appropriate in response to that signal. So the function of the effector is to provide a physical sensation. So when I defined the structure of the membrane in trying to provide the audience with what it really represents, I defined the membrane switch we've been talking about as an awareness of the elements of the environment through a physical sensation. What was totally interesting was this is the exact same definition used for a particular word that was already in the dictionary that most everybody was familiar with. The word is perception. This is the exact definition of perception, an awareness of the elements of the environment through a physical sensation. And all of a sudden, I called the switch perception, and all of a sudden everybody in the audience started to understand right away what I was talking about. Perception controls behavior. The switch, the awareness of the elements of the environment, then engaging the effector, which sends a physical sensation, connects an environmental signal to a biological behavior. The switch is called perception. And therefore, when I told the audience, perception controls behavior, everyone immediately understood what I said. And what was really interesting is by the time we really get to this part of the discussion, and we're talking about behavior being controlled, you might realize something. We haven't even talked about DNA. The issue is, life isn't controlled by DNA. DNA is a blueprint to make the proteins, but the proteins aren't controlled by the DNA. The proteins are controlled by the signals of the environment, which are mediated by the perception. So the first real hard conclusion in the nature of life in the study that we've been talking about right now is perception controls behavior, which simply implies an environmental signal is picked up by a switch called perception, which then directs a signal to a protein mechanism inside the cell that creates a biological function that will respond to the signal in the environment. So all of a sudden it's like, wow, life isn't controlled by genes. No, it's controlled by the signals in the environment. And this is very interesting because I observed this in my cultures of stem cells. That in creating the culture of stem cells, one of the first things you do is digest the tissue to free up the cells. But in the digestion process using enzymes, the enzymes clip off the receptor proteins. And so as I'm preparing my cell cultures, one of the first things that happens is the receptor proteins are lost from the surface of the cell. The cell can't read the environment, for at that moment the receptors are defective. Interesting point. When the cell cannot respond to the environment, it remains as a little round ball. It doesn't express any behavior whatsoever. That the cell does not express behavior until such time that the metabolism occurring inside the cell leads to the replacement of the receptors that were lost by the enzymes. Once the receptors start to appear back on the surface of the cell then engages in behavior. So in a very interesting point, when the cell cannot respond to the environmental signals, the cell does not express behavior. The signals and behavior are tightly integrated upon one another. So now we've gotten to the point. Behavior, yes, controlled by the environmental signal, and behavior is created through the action of the proteins in the cytoplasm. But then here comes an interesting point. As I said, there are over 100,000 different proteins. A cell does not make all of the proteins all of the time. It makes the proteins that it needs to use. Well, consider this possibility, that a cell is in the environment and a signal shows up that wasn't there before, and the cell has to make a response, but now it doesn't have the proteins in the cytoplasm to make that response. And yet the signal is at the door knocking on the perception switch, saying, okay, I'm out here, and the cell has to make a response. Now the question is, what does the cell do when it doesn't have the protein? Aha! This is where we bring in the genes, because the genes are the blueprints to make the proteins. But the question is this: how does a gene know when it should offer its blueprint to make a protein? The genes don't see the world. The genes are encapsulated and enclosed within the nucleus and have no visibility or awareness of what's really going on in the world. In that regard, genes don't know what should be activated and which proteins should be made and which ones should not be made themselves. That the genes are not self-actualizing. And this is very interesting because if we talk to conventional biologists or read the current literature, we always hear phrases such as a gene turned on, a gene turned off, giving us the impression that genes are intelligent units, that genes have the ability to respond to the world that they live in by observing that world and making responses through an intelligence, something like a self-actualization. The interesting part is, these are all words based on the original belief from the central dogma that genes control life, that genes are the active component. That was perceived in the dogma. And ever since then, we bought into the concept of genetic determinism. Genes are controlling life. Every time we invoke the concept that a gene expresses control, we're suggesting that the gene is able to self-actualize, that it has a consciousness or an intelligence to it to allow it to turn on or turn off. Well, this, as I said, is what we bought from our belief that genes were actually in control. But the reality is, genes are not in control. We have already demonstrated that by the fact that we could remove the genes from a cell and the behavior is still there. But now we can understand why that occurred, because the proteins are in the cell, and the proteins are controlled by the perception switches, and therefore the genes weren't even necessary, and that's why enucleating the cell didn't really cause a problem. However, the cells ultimately did die when the nucleus was gone. And the question is, well, then what was the role of that nucleus? And the answer is simply this. The genes provide the blueprints to make the proteins that comprise the machinery of the cell. That if I remove the nucleus, I remove the blueprints. The cell can still go along as it has the proteins. However, if a protein breaks down, which they normally do through wear and tear, just like any machine, when the proteins break down, I need to replace them. If there's no nucleus, then there are no blueprints. And so in a nucleated cell, when the proteins break down, there is no recourse to identify how to make a new protein. And when enough proteins break down that interfere with the vital functions of the cell, that's when the cell will begin to die if it doesn't have genes. So the cells die not because the nucleus was removed and interfered with the operation of the cell. The cells die when they're enucleated because at some point down the line, when I need to replace a protein that is vital for the function, I don't have the gene blueprint to do so. The cells without a nucleus can't reproduce their parts or themselves. So interesting point is, removing the nucleus prevents reproduction. And when you understand that, you realize, my goodness, in all the books we've given the nucleus the responsibility of being the brain. But in truth, the nucleus is just the gonad. The nucleus is the reproductive element. Now, if we look at the nucleus as a gonad and how it applies to reproduction, then we go back and have to say, let's look at those chromosomes, because the chromosomes contain the DNA where the blueprints are. Well, the interesting part, if you can recall from the earlier discussion, is that chromosomes are made out of 50% protein and 50% DNA. And in 1944, when Avery McLeod McCarty revealed that the DNA was the element that controlled the traits, protein biochemists lost out. Nobody cared about the proteins. Everybody wanted to deal with the DNA. So all subsequent experiments from the late 40s onwards really involved this: isolating the genetic material, the chromosomes, from the cell, separating the components of the chromosomes, the proteins, from the DNA, saving the DNA to do the molecular genetic experiments, and throwing away the proteins. So for 50 years, we threw away the proteins, which constitute about 50% of the nucleus. At some point, people started to say, 50% of the chromosome is protein. That's a lot of filler material. Is the protein doing something? And people started to look at the proteins. And all of a sudden, they found, oh my God, we didn't understand this because we've been throwing away the proteins for 50 years. There was a new understanding of the relationship between the proteins and the DNA. In the experiments that revealed that DNA was self-regulatory, those were experiments that were done with pure DNA. They in no way reflected what was actually going on inside a living cell where the DNA was already complex with the proteins. When we put the proteins back with the DNA, we get another understanding. It changes our belief in what we describe as genetic control and genetic determinism. It goes like this. Up to now, we've been talking as if they were controllable elements that genes turn on and turn off. And the truth is, a gene is simply a blueprint. that as a blueprint, it has no on and off, just as much as an architect's blueprint doesn't have an on or off. The blueprint is not self-actualizing. If it were, the architect would only have to throw the blueprint at the building site, and the self-actualizing blueprint would build the building. And yet the reality is, no, a blueprint is just information. Now the question is, what activates the information? Well, in this case, the contractor who reads the blueprint and then decides to act upon the blueprint. Well, when we go back to the cells and the DNA and we say the DNA is the blueprint, but what reads it? What's the contractor that is involved with activating the expression of a gene? It turns out it was the protein that we threw away for 50 years. All of a sudden, there's a new insight in understanding the nature of the chromosomes. Chromosomes are actually DNA strands surrounded by a sleeve of protein. Basically, what this says is as follows. The blueprints to make the parts of the human body are encoded along the length of the DNA. Each blueprint is referred to as a gene. But the genes cannot be seen inside the cell because when the DNA is in the nucleus of the cell, the DNA is covered by a sleeve of protein. If you want to read that blueprint, you have to remove the protein first. This was not recognized before, especially in experiments where you throw away the protein and just study the DNA. When we go back to the living system, we realize that in order to get to the blueprint, you have to remove the protein sleeve. To control a gene, then, is actually not controlling the gene, but is controlling whether the sleeve is on or off. Yeah, but now you already have some insights as to how can I control the sleeve. And it goes back to this. Remember, the sleeve is protein. That protein can change its shape when it responds to environmental signals. So consider this. In the resting state, when we're not reading a gene, the protein sleeve is covering up the DNA, and the gene is not active. You can't read it. It's underneath the protein sleeve. But now we go back to that situation I described. A signal shows up in the environment, is read by the receptor of the cell, and the cell wants to engage a behavior, but can't do it. Why? The proteins aren't present in the cell. So what we need to do is we need to get the blueprint to make the protein. Well, here's how it works. The signal from the cell membrane then travels to the nucleus. And in the nucleus, the signal complements with the specific proteins that cover the gene that we want to use. So when the signal binds to the chromosomal proteins, it causes the sleeve to pop off. When the sleeve pops off, the blueprint's exposed. Then the cell makes a copy of the blueprint as it's exposed. The copy is called RNA. The RNA is the actual physical blueprint that leaves the nucleus of the cell and goes into the cytoplasm of the cell. In the cytoplasm of the cell, there are protein devices that will read the RNA blueprint and create the new protein. So basically, it says this. Genes are neither on nor off. In truth, genes are either covered or uncovered. But the cover and uncovering is the character of the protein. And therefore, if you want to use a gene, you actually have to regulate the protein sleeve. But the regulation of the protein sleeve is controlled by signals that are sent by the membrane from perception switches. So all of a sudden, we go from a biology where we looked at the genes as these devices that control their lives. People think that if they walk down the street someday, one of their genes is going to turn on them, and this gene is the culprit, that this gene caused a cancer. When it turns out, no, that's not actually how it works. The gene can only be activated when it's in response to an environmental signal mediated by the switches in the membranes, which are called perceptions. So when we look at this and put it all together, it says that the environmental signal via the perception switch sends a signal to the nucleus to uncover the gene to allow us to make a copy of the gene and then make proteins derived from that gene. What this basically says is genes control nothing. Genes are responsive to the signals of the environment. This new field of study is something called epigenetics. And in fact, this is what I was studying back in 1977, even though epigenetics is a term that is probably no older than 1995. Almost 20 years before the field of epigenetics was established, I was already seeing the action of epigenetics in my clone cultures. How was that? Yes, put my culture in condition A, environmental condition A, the environmental signals selected the genes to form muscle. Take the same cell and put it in environment B, and the environmental condition in that culture selected a completely different set of genes and caused the formation of bone. And thirdly, a completely different set of environmental signals leads to the activation of genes that provide for fat. The control of gene activity by environmental signals is the new science of epigenetic control. Well, most all of us and the students I was teaching in medical school are very familiar with a concept called genetic control, which literally means control by genes. The new understanding of epigenetic control is just barely making itself into the public domain at this time. It's still an identity of a mechanism that is understood at the leading edge of science, and even most scientists still don't understand the concept of epigenetics. Epigenetics is the mechanism by which we control genes. Now, the interesting aspect about epigenetics is mediated by environmental signals through perceptions. So rather than the old belief that everyone bought into that genes control life, and this is the big problem for the Human Genome Project, because when it was revealed there were only 25,000 genes, the reason why that was a failure to them was that it obviously takes more than 25,000 genes to make the complexity of a human. And so they didn't understand the control mechanism because they believed the genes were controlling themselves, and there obviously weren't enough genes. Now we find that epigenetics exists. Epigenetics is, in fact, the equivalent of a second genetic code. It's not secondary genetic code, it's actually a primary genetic code because epigenetics can change the readout of the genes. Well, we used to think one gene makes one protein. With studies on epigenetics, we find this. By the diversity of environmental signals, there's a diverse response that can be manifest from each gene. In fact, we now know this, and this is an outstanding fact, that through epigenetic mechanisms, a single gene can provide for over 2,000 different proteins, because epigenetics is the reader of the blueprint. Epigenetic mechanisms can cut the blueprint into pieces and splice it together in a different configuration and get a completely different readout from the same blueprint. Epigenetics can only read part of a gene and ignore the rest of the gene blueprint, again changing the product. And all of a sudden, there's this new genetic code that starts to come into our understanding, a genetic code that is so powerful that it is a contractor that can read the genes and change the structure of the organism. Well, here's an interesting fact. The genes in a human are virtually identical to the genes in a chimpanzee, which are virtually identical to the genes in a rat. All of us have the same kind of building blocks. But if we all have the same genes for the same building blocks, why don't we look the same? And the answer is because how the organism is constructed is not based on the genes that make the building blocks. It's based on the epigenetic control. That humans have a different epigenetic readout of the building blocks than a chimp. So we can use the same building blocks and somebody can create a chimp, and using a different way of assembling the same building blocks you can create a human, and even further altering the assembly of the building blocks you can actually provide for a rat. And so it turns out that the control of life is not built into the genes that provide for the building blocks, the genes that we have studied from the beginning of the understanding of the genetic code to the very moment. That's where most research is involved. However, the field of epigenetics is changing our emphasis. We're now beginning to recognize that rather than messing with the genes, it's epigenetics that can alter the readout of the genes. Interesting to this point. Most of us are even missing genes, and yet we don't even know that we're missing the gene. The reason is because via epigenetic mechanism and a process called genetic capacitation, other genes can be modified by epigenetic mechanisms to provide products that would have been derived from the missing gene. So you would never even know that you were a missing gene. Epigenetics is this global mechanism that covers the readout of our biology. And the fundamental understanding behind epigenetics is controlled by environmental signals. Why is this important? Because in our history to dominate and control nature, we thought the genes controlled life. And if I want to change the character of life, I have to change the genes, genetic engineering. And what we're beginning to find out is now, my goodness, wrong emphasis, wrong genome. We should be looking at epigenetics because epigenetics can alter the readout of any particular gene. But you can change epigenetics not by changing the genes, but by changing the perception to the environment. But wait, how did perception get in there? And then go back and realize the environmental signal that controls epigenetics is mediated by the switches on the cell surface called perception. And all of a sudden we start to see, yes, how you perceive the world now not only controls just the biological proteins that make the functions of respiration, digestion, movement, but how you see the world, your perception of the world, selects which genes are going to be activated, and not only just selects the genes, but can modify the readout of the genes to make a protein that would best fit the environmental circumstances of the time. And then when you realize it, you say, oh my goodness, think how much easier it is to change our lives not through genetic engineering, the old way of messing with the protein-building genes, but by changing our perception, which then alters the readout of our genes. And in fact, when I talked about the influences that cause disease, and I mentioned trauma, toxins, and thought becomes the big element in this regard. Since perception runs our genes, misperceptions can misrun our genes. And misperceptions are interpretations of the world that are inaccurate. And yet the reality of the biology, as I mentioned, is it responds to perceptions. So I could call this the biology of perception. And yet, why did I call it the biology of belief? And the answer is, because not all of our perceptions are true. Some of them are totally inaccurate. And yet, even if they're inaccurate, they still run our biology. And inaccurate perceptions will inaccurately run our biology, which would lead to dysfunctions and disease. And that is why thought becomes a prominent contributor to the state of health that we express. So it then recognized the role that perception is the primary mechanism that controls biology. When we start to look at how perceptions influence cells, I started to get some very fundamental nature of the degrees and types of perception by observing my cells. I found that I could actually subdivide perceptions into 3 categories based on the responses of the cells. That I would call things that I would put into the environment that the cells liked, that supported their growth, as positive growth-promoting perceptions. Meaning, if I put nutrients in a petri dish, the cells would read that perception of the presence of the nutrients, and the cells would move toward the perception. So I found that any perception that causes the cell to move toward it represents a perception associated with growth, because growth means to take in that thing, whatever it is, and assimilate it into your life. In contrast, if I put some things into the culture that actually threaten the life of the cells, such as toxins, they responded to the perception of the toxins with a completely different response. That when toxins were put in the petri dish, the cells didn't move to the toxins, they moved in the complete opposite direction. And then I started to realize very simply this: if things were in the environment that threatened the survival of the organism, in response to that perception, the organism moves away from the stimulus. So growth stimuli cause the organism to move to it, be open in the process, and take in those stimuli to incorporate them in the growth of the organism. And those stimuli that required protection caused the organism to move in the complete opposite direction, away from the stimuli, and in the process close themselves down to protect them from the noxious environment that they found themselves in. So I had 2 fundamental responses or perceptions, those that promote growth and those that lead to behaviors that we would classify as protection behaviors. There is indeed a third response to perception, which is actually no response at all, meaning that some things in the environment are totally innocuous. They don't promote growth, but they don't kill you either. I usually refer to these innocuous things as elevator music. You get into the elevator, you don't actually dance, but it doesn't kill you. It's just background. So now I say organisms respond to their perceptions of the world. That the types of perceptions could be categorically divided into 3 different classifications. Those perceptions that provide growth, and these perceptions cause this organism to move toward the stimulus and take it in. Those perceptions that require protection responses, which cause the organism to move away from the stimulus and close themselves down. And then those perceptions which are just elevator music, background, innocuous, and no response is required at all. So it's interesting, it says that there are basically 2 fundamental actions that can result from a perception: growth or protection. What I really observed that was very important in understanding the lessons the cells were trying to teach me was simply this: that cells could not be in growth and in protection at the same time. They were mutually exclusive behaviors in this regard. Growth, move to a stimulus, be open to assimilate it. Protection, completely opposite. Move away from a stimulus, close yourself down to protect yourself from the environment. What's interesting about it is the simple, obvious nature that a cell cannot move in both directions at the same time. Cells can be in growth, where they're actually responding to their environment and building themselves up, or cells can be closed to the environment in protection, walling themselves off, but you can't be open and closed at the same time. You can't move to a stimulus and away from a stimulus at the same time. So I started to recognize that living organisms were responding to the stimuli in regard to their behavior, and that could be described as either responding to it in growth or responding to it in protection. Well, it's interesting because when we bring this up to the level of human biology, here's an interesting fact: that growth and protection, the extremes of stimuli between growth and protection as perceived by the human, can be described as love and fear. Love is the ultimate growth response for a human. Once you experience love in your field, if you perceive love, you will move to that love no matter where it is and try to get it and assimilate it and take it in. If love's on the other side of the mountain, you will climb over that mountain to get to love and take it in. If the mountain is too high, you'll dig a tunnel through the mountain to get to love. It is the ultimate growth perception, love, for humans. In contrast, fear is the exact opposite. When you fear something, you try to get away from it. You try to close yourself off. You wall yourself off from fear. You get in a protection response. So that it's interesting. Well, yes, many things can be supporting our growth, our nutrition, the environment that we live in, the physical house, and all these other kinds of things. It turns out that love seems to be the most profound of all of the perceptions for growth. It is interesting because in Eastern European countries, there are many, many orphanages, because in those countries, people have children as promoted by the church, but then they recognize when they get past a certain number of children they can actually maintain, all the extra children are dropped off at the orphanages. Well, it's interesting because the orphanages provide interesting study sites. When you observe the kids in these orphanages, you observe something such as this: they all are fed very well. They all have clothing, they have their rooms, they're protected from the elements, they have everything except love. And the interesting part about that is, in the absence of love, every one of their vital statistics are suppressed by 30% or more. Their physiological correlates are greatly depressed. They're really very sickly in general. Many of them are autistic. Now, autism in this particular situation is really derived from the fact that if they're not getting love, they're not being allowed to open themselves and expose themselves to the outside world. In the absence of love, they close themselves down. Autism, in this particular situation, is a result of closing themselves down, protecting themselves from the world that doesn't offer them that love. So it's very interesting that there is a correlation in the children of how they respond to love and how their physiology is a direct connection to their perceptual experiences. Well, it turns out why these perceptions that support us give us health, and why our perceptions that where we buy into fear and threats of our survival actually cut down our life is related to a mechanism in the human body called the HPA axis. The H stands for hypothalamus, the P stands for pituitary, the A stands for adrenal glands, and they collectively interact in the following way. The H, the hypothalamus, is the portion of the brain that does the interpretation of the perceptions. So when you observe your world, your hypothalamus is designating whether this perception is one that promotes growth or this perception requires a protection response. When the hypothalamus perceives signals in the environment that require a protection response, it must engage the body's behavior to deal with the requirement of protection. So this is why the hypothalamus, upon interpreting a stress response, will send that information to the pituitary gland. Well, the reason why the pituitary gland is involved is very simple. As you may remember even from elementary school, the pituitary gland is called the master gland. It is the gland that sends signals to 50 trillion cells to coordinate all their behaviors. So if I'm going to direct the operation of the machine, the pituitary gland becomes a conduit to get the information to all of the cells of the system. So the situation of a stressful existence interpreted by the hypothalamus is forwarded to the pituitary gland. But since the threat comes from the outside, and I need to engage a protection response, this is why the signal is then forwarded to the adrenal gland, because even the adrenal gland, if you remember from high school classes, is the gland that's associated with fight or flight. The adrenal gland is the gland that engages the behaviors of protection. So when we see a stressful environment before us, we activate the HPA axis, which results in our getting into a protection response. Remember, protection is closing yourself down and walling yourself off from the noxious environment. In this part, what do the adrenal glands do to promote this behavior? And this becomes very important for the following reason. The adrenal glands send out stress hormones. Well, the stress hormones inform the system that the threat that we may experience is coming from the outside world. Since we're going to have to deal with the outside world, we really want to nourish our arms and legs, because in a fight or flight behavior, those are the organs that we're going to use to maintain our existence. So in fight or flight, using arms and legs, I want to nourish those arms and legs and give them all the energy I possibly can to carry out their very important task of saving my life. So to do that, the body shifts the allocation of energy to the protection system. Well, in the textbooks of physiology, they say this in response to the fact that when stress hormones are released into the system, the blood is preferentially sent to the arms and legs. Well, that makes obvious sense. I want the blood, which carries nutrition, to go to the arms and legs so I can energize my muscles and allow me to use fight or flight behavior to protect myself. But when you go back to that sentence I just used, there's a very important part of the phrase. The phrase goes, the blood is preferentially sent to the arms and legs. That causes us to pause a second and say, so where was the blood before it was in the arms and legs? And that's when we realize the blood was in the viscera, the gut. But what's the function of the gut? The blood is nourishing the visceral elements to do what? Well, the function of the viscera is growth and maintenance of the body. All the organs of metabolism and digestion and energy distribution and filtering systems and all the things that keep us healthy and alive are really the elemental functions of the viscera. And yet, when I'm in a state of protection, I need to send all my nourishment to my arms and legs. And this is accomplished by the fact that when we see an environment that requires protection, we gauge the HPA axis. When the stress hormones are released into the body, the stress hormones cause the blood vessels in the viscera to constrict. When the blood vessels are constricted, the blood flow is restricted. No longer is blood flow easily going through the visceral elements. They stop their functions basically because without blood, there's no nourishment. Then the processes of growth are inhibited. That growth mechanisms are actually shut off when we are in a stress response because we're allocating all the energy via the blood to the peripheral organs. Well, in saving energy, we also shut down some very, very vital, important systems as well. One of those is the immune system. The immune system and the adrenal system are both actually protection systems, but they protect us from different things. The adrenal system protects us from threats outside of our body. The immune system protects us from threats that enter into our body, threats such as virus or bacteria or parasites that would like to take over the operation of our system and feed off of us. These elements are inhibited or restricted through the action of the immune system. But if I recognize that my stresses are more important in the outside world than the stresses I perceive in the internal world, that's when I'm activating my HPA axis. That's when I'm releasing the adrenal stress hormones, and the stress hormones are encouraging protection from outside threats. Well, to allocate all the energy it can, the system will inhibit the action of the immune system. The body recognizes the immune system uses tremendous quantities of energy to carry out its protection functions. Yet if I'm running away from a saber-toothed tiger and I'm having a bacterial infection, my central nervous system, my intelligence system will easily recognize that perhaps we ought to put all the money into running away from the tiger, and if we survive, then we can actually go fund that immune system. Well, this is actually what happens, because when we activate the adrenal hormones, the stress hormones don't only cause the blood to go from the viscera to the periphery, the stress hormones also shut off the immune system. Interesting, this is a fact that has been known by the medical profession for a long time, and they actually use stress hormones very specifically for this function when they're busy transplanting organs and tissues into a recipient. Because the organs and tissues of the transplant are foreign tissues, the recipient's immune system would immediately set about to eliminate the foreign invasive transplanted cells, the tissues and organs that the doctor provided to the recipient. Well, if we don't want the immune system to reject the organs, then what you would really like to do is restrict or reduce or inhibit the immune system. This is exactly why recipients of grafted tissue receive stress hormones, because when they receive them, it inhibits the activity of their immune system and allows the graft to take and allows them to maintain the foreign tissue. Yes, but this is a very unique biological consideration in an experimental situation at that. What does it mean in regard to stress hormones in our everyday life and the presence of these stress hormones interfering with our immune system? And it goes like this, that all of us are right now infected with almost all the viruses and bacteria and pathogens that affect humans. I could take a blood sample out of every one of you right now, no matter how healthy you feel. I will show you that you have organisms in your blood. Well, it's very interesting because biologists are very familiar with these organisms. In fact, they call them collectively opportunistic organisms, meaning this, you give them the opportunity and they'll take over. If you are in a state of good health and a state of growth, you are supporting the operation of the immune system, and the immune system will keep a lid on these invasive organisms. While it might not be able to kill all of the organisms, it certainly can restrict their growth and limit their proliferation. And this keeps a lid on those opportunistic organisms from really impacting us. However, once we start to get into a stressful environment, then what happens is the stress hormones from the adrenal glands start to inhibit the immune system. When the immune system operation is interfered with, these invasive organisms then get the opportunity to start proliferating and taking over, which basically says this. Most people, when they're in a happy state of life, are very, very healthy. But once they start to get stressed, they start to get sick. Some people say, well, I just caught something when I was stressed. But the truth is more likely you already had something, and now that you're stressed, you remove the cover of the immune system by inhibiting its action through stress hormones. So all of a sudden you start to realize the very debilitating character of stress, that stress shuts down our growth mechanisms. The longer we're under stress, the more we're inhibiting the action of the viscera. That stress shuts down the immune system because the more we're concerned of fears from the outside, the more we reduce the function of the internal protection mechanism just to allocate its energy to be used for the external threat. And well, if that's not bad enough, there's actually what I refer to as the insult to injury. There's a third character of the stress response mediated by the HPA axis, and that is the following. Think about it this way. If you are in a fight or flight response, do you think you would rely on reflex behavior or conscious information processing to deal with the fight or flight response? The answer is obvious. Reflex behavior, because it's absolutely faster and more powerful and able to control us rather than conscious thinking. conscious thinking is a slow process. While you'd be sitting there as your car is going out of control in your middle of an accident, if it was controlled by consciousness, you might hear yourself going, whoa. Rather than relying on that intelligence to run the car, when you're in a stress response, the stress hormones cause the blood vessels in the forebrain, the conscious part of the brain, to constrict, just as the stress hormones causes the constriction of the blood vessels in the viscera. And just as when the visceral blood vessels are constricted, visceral function stops because it's not being nourished by blood, in a stressful situation, when we constrict the blood vessels in the forebrain, we inhibit conscious thinking. But in the process, the blood is then preferentially sent from the forebrain to the hindbrain, where we're really nourishing the reflex centers. So when we're in a stress response, we primarily operate from hindbrain reflex mechanisms and inhibit forebrain consciousness, which in the end simply says this: when we're in stress, we're less intelligent, because at that point, we're not using intelligence, we're just using reflexive behavior. So these are three very big detriments to our normal physiology. But this isn't a detriment of the way evolution designed the system, because evolution designed the system only to use the stress response at certain periods and only for short periods of our lives. To run away from that saber-toothed tiger, for example. Once you got away from that tiger, then you automatically go back into the growth mechanisms to nourish yourself and to maintain your life. If another tiger shows up, you run until you get away, and then you go back into the growth mechanism again. So basically what the information really reveals is this: under normal evolution, a stress response is usually a very restricted response, only used for short periods of time. But if you look at the world that we live in today, you see we live in a world where stress is the key word. Everything is stressful. The jobs are stressful. The environment's stressful. The food's stressful. What we're beginning to realize is while evolution designed the stress system to be only used for short periods of time and short spurts and pulses, we are now operating from stress, most of us, 24/7, 365. This has caused a great debilitation of health of the individual and of the population. We see it throughout the entire population in regard to the crisis in healthcare of so many people being sick for so long. And the fact is, in our old perception, the perception that is actually conventional allopathic medicine today, we look at a sick body as a Newtonian physical mechanism whose chemistry is off, and that we can fix a sick body by just throwing in drugs. However, the new biology based on perception and epigenetics reveals that it's not the machine that's wrong, it's the environment and our perception of the environment that is caustic. Interestingly, it takes us back to that very first day I did a cell culture and exactly what Irv Koenigsberg warned me of. He said, if the cell isn't healthy, before you try to change the cell, look at the conditions of the environment. And this is exactly where we should be today because, as I said, we are cells incarnate. Even though we're comprised of 50 trillion cells, we actually represent a composite of 50 trillion cells living as one giant cell. And the significance is when we get sick, while we have a tendency to say, well, let's fix your physical body, the reality of Irv's warning comes to play here and says, before you fix that physical body, first consider the nature of perceptions, because perceptions are the things that primarily distort the normal functioning and operation of the machine. And I refer back again to the fact that I said the operation of the biology is due to the signals and the proteins. And then remember, only a very small percentage of people can claim that any of their dysfunctions in life are due to the proteins, that then the vast majority of us have to recognize it's the signaling that is a responsible element. Just as Irv said, it's the signaling from the environment, our perception from the environment, that controls the status of our health. Yet, while this is very interesting information, in the end it is most empowering because, again, it says, if you really want to control your biology, interior health, as well as your exterior reality, you have to recognize the power of your perceptions in shaping both your physiology and your life experiences.
This concludes CD 2 of The Biology of Belief. Our program continues with CD 3.